Okay, I think we'll get started to keep on time. I'm Alan Landay. I'm the chair of this session, Restoring Immunity and Aging Gracefully, which I hope we all are aging gracefully here in Amsterdam, enjoying the hot weather, but again, a great environment for this meeting. What we're doing today is, this is our poster discussion session, so we have four discussants and posters to present. I'd like to have interactive discussion after each of the presentations so that we can really provide some good insights to the presenters and understand more about the integrated aspects of the topics that we'll be covering today around telomeres, glycans, the microbiome, and immunometabolism. So I think fairly diverse topics, but all are highly relevant to HIV pathogenesis and clearly to the aging agenda. If I could then, I'll ask our first presenter, Andrew Sue, to come up, and his title is Shorter Cell Subset Telomeres in HIV Slow Progressors than in HIV Non-Slow Progressor Women. Anthony? Sorry.
Let me click on this here.
Yeah, click on that. Your slide should come up.
Great. Let's see here. Okay. Oops. So I want to start by saying that I really like the title of this session a lot, Restoring Immunity and Aging Gracefully, because it uses such elegant language to describe our research, which is a world that I perceive through flow cytometry plots and multivariable models. I don't know what the intention behind the title was, but to me, the language implies that we're not trying to stop or even slow down aging, but rather to minimize discomfort and maximize dignity. That we're not trying to mold an invulnerable immune system, but to restore it to its proper function. The most direct implication, perhaps, is that restoring immunity is indispensable to the pursuit of aging gracefully. I know I'm spending a lot of time setting the stage and waxing philosophical, but I think it's important in the context of this talk to interpret the data in the context of natural aging instead of merely comparing between HIV groups. And also, I need to grab your attention now because I'm actually showing flow data in a little bit and I don't want anybody to fall asleep. Just as restoring immunity and aging gracefully are inexorably linked, so too is the link between HIV infection and accelerated immune aging. I don't have time to go over all of this in more detail, but roughly speaking, HIV infection forces the immune system to maintain a constant state of activation. Here, I present an interim analysis of a study that aims to characterize immune aging in the context of HIV within relevant immune subsets. The design compares between three age-matched groups: the HIV-positive non-slow progressors, who are all on CART, all with undetectable viral load; the HIV-positive slow progressors, who, with varying extents, can control the HIV virus without CART; and the HIV-negative controls. And I want to quickly pause on the slow progressors. These are not all elite controllers. They have varying abilities to control the virus. And I use flow cytometry and cell sorting to sort the following cell populations: CD4 T cells, B cells, and proliferative and senescent CD8 T cells, within which we measure telomere length. And as many of you know, telomere length is a marker of intracellular immune aging. The more times a somatic cell divides, the shorter the telomere length. I also use flow cytometry to track the CD4 CD8 T cell lineage between the HIV groups. So first, let's take a look at the telomere length within the four immune compartments. As you can see, there appears to be no detectable difference between groups in the CD4 T cell compartment, whereas in the proliferative CD8 T cells in the bottom left corner, the difference exists between those living with HIV and those without HIV, the HIV-negative controls. There's no detectable difference here between the progressor groups. In contrast, in the B cell and senescent CD8 T cell compartment, there appears to be the greatest difference, not between the HIV-negative controls and those with controlled infection, but between the slow progressor and the non-slow progressor groups. These data may reflect the greater proliferative burden placed on the CD8 T cell group in HIV infection. So next, let's look at CD4 and CD8 T cell lineage. There's a lot of colors and lots of data here, so I'll just briefly describe how to interpret this. On the left, we have the CD4 T cells. On the right, we have the CD8 T cells. Across the rows, we have the 3 different HIV groups. And I've connected the cell populations within individuals within each of these groups. And so when you see a blue line, that means the next subsequent level of differentiation had a larger population within the same individual, and vice versa can be said about the red lines. You can probably see immediately that, again, no difference exists within the CD4 T cell group. And in the CD8s, the non-slow progressors and the HIV-negative controls share a fairly similar profile, whereas in the slow progressor group, there appear to be less naive CD8 T cells and an expanded effector memory cell population. So further reinforcing the same point here, if we look at the CD4 and CD8 T cell ratio, there's no difference between progressor groups. It's as though people living with HIV are just have less CD4 T cells compared to CD8 T cells, which is rather well known. But within the CD8 T cell compartment, there appears to be a progressive staircase-like pattern where the slow progressors had less proliferative CD8 T cells or were older in their immune system compared to the non-slow progressors and in turn compared to the HIV-negative controls. So to conclude, I just want to summarize by saying that everywhere we looked, it seemed like the slow progressor group presented a more aged immune profile compared to even the controlled HIV-positive non-slow progressors. And in the beginning, I wanted to talk about the context with natural aging, and although it seems like the investigation of HIV slow progressors is an issue of the past because nowadays we strive to treat everybody with CART regardless of their progressor status, I think these data still shed light and provide valuable insight on how the non-slow progressors may age over a longer period of time, as well as what the immune system does to try to control HIV in the absence of CART, and how similar those processes are to the world of natural aging. So with that, I'd like to thank all study participants, collaborators, and staff, and I'm happy to take any questions.
Thank you. As I said, we'll take questions. Are there specific questions for Anthony at this point? Yes, microphone 2. Please identify yourself.
Yeah, Jose Rivas from Madrid, Spain. Thank you for your presentation, Anthony. 2 questions. One, this is if I understood correctly, this is cross-sectional, right?
That's right, yeah.
Do you have sequential data? You're going to have sequential data to see what's the evolution of telomere length in each subset comparing the groups?
We've designed the study so that in the near future, we can collect longitudinal data and make those comparisons. That's probably going to be outside my PhD, but it'll be done.
Yeah, and the other question is, can you tell us about, this is slow progressors, but you said that there are some elite controllers and non-elite controllers. What are within that group, the drivers of senescence were those who were not elite controllers?
So regarding these data specifically, I don't think that the elite controllers were driving because of the 35, only 4 were elites. And so, like I said, this is an interim analysis. We plan to more than double the power very soon. And when we do, we can do a sensitivity analysis within the elite controller group to see, where the effect truly lies. But, 31 out of the 35 here are not elite controllers.
Hi, Peter Hunt from UC. Oh, sorry. Was there someone?
Yeah, go ahead.
Yeah.
Peter Hunt from UCSF. Really nice presentation, and congratulations on sorting the CD8 T cell subsets, which is important to sort of shed light on the pathogenesis of what's happening. And one possibility for the somewhat unexpected results at the slow progressors is that there's a proliferative and differentiation defect in effector CD8 T cells in HIV, such that they don't fully terminally differentiate and undergo multiple rounds of cell division and shorten their telomeres. That's sort of what a normal effector CD8 T cell is supposed to do. And there seems to be a defect with that in the context of HIV, particularly in people with more advanced disease. But one question I had with the HIV negative comparisons was this all restricted to CMV seropositive individuals? Because that's that's a factor that profoundly affects CD8 T cell differentiation.
Yeah, so and I want to quickly comment on the first. That's a really interesting insight that I had not considered, but it may describe that sort of traffic jam pattern in the effector memory T cells in the non-slow progressor compartment. Our serology data is incomplete. I know that in the cohorts that are here, most people are co-infected with CMV, but that we're in the process of gathering serology now for HCV and stuff like that. So, yeah, thank you. Are we on? Okay.
Thank you. Yeah. Microphone number 2.
Yeah.
Go ahead. It's you.
Okay, I'm Robert Reinhardt. I think I represent old people. And nice talk. And I was just wondering, I hope this makes sense. Do you think it's worth also exploring whether there are reservoir differences between these slow progressors and non-slow progressors because of the effector memory and central memory differences that you observed? And I take it you haven't gotten there yet, but is it worth exploring?
I think it is, and okay, I'm going to spend 30 seconds to talk about this in a bit. I've always, we've always had this model that it's because of the sustained immune activation, like I alluded to in the beginning. So, in the beginning, the T cells proliferate, they activate, and then they kind of die down after a while as the infection reaches latency. And so, we've always thought that maybe it's the area under the curve that represents the usage of the finite proliferative potential of the immune system. But if that were the case, then we'd expect to see strong correlations between aging and the time since infection, which we don't. But maybe that data is still a little bit in its infancy. But because we don't, one possible explanation is that the amount of proliferation that's happening in the acute infection stage is so much more that it doesn't really matter how long that they've had the virus subsequently. And one of the things, to your point, is that depending on how the acute infection is being treated or managed or what other factors are existing at the time, the reservoir can differ greatly. So I think it is worth definitely looking at the diversity of the reservoir between these groups, but maybe also within the umbrella investigation of what is happening during acute infection. Thanks.
Any additional questions or comments from the audience? Maybe I could ask one. In the subset analysis from the original paper that Janice Georgi and Rita Efros published, and that's actually probably an old paper by now, it's 23 years ago almost, that original paper, they looked at CD28 negative cells among the 4s and 8s and did show differences among HIV. So I'm wondering if you've looked in your populations around that question of subsets among especially the CD8 cells and populations.
Right, so it was actually one of the factors that led us to choose CD28. It's kind of a dated marker. There are other ones to use nowadays.
Right.
But the main difference between that study and ours is that it was the people were not treated, and it wasn't clear whether they were able to control the virus or to what degree they were able to control the virus. It may be that they would have been non-slow progressors, but that the virus was just rampantly, replicating in those people. So it's a little bit of a different study design, and that perhaps explains the difference between our results.
And one point on elite controllers, since they may have slightly higher levels of virus, and we're now doing studies in a variety of venues and treating elite controllers, how do you think virus itself might be driving the actual telomere shortening, and what would you predict if you were looking at elite controllers treated on therapy, since they also show CD4 decline, how that might affect telomeres?
So, sorry, the elite controllers have more virus?
They have a small amount of virus, low-level virus compared to heart control.
Right,
that's been published by a number of studies.
Right, they have less virus.
Yeah,
right?
they have a single copy, low levels of virus. Yeah.
Yeah, there have been a few papers, I think, correlating telomere length depletion within specific cell compartments in the context of how much virus is present, and I'm not sure what the direct relationship is. My speculation would be that it acts through the aging immune system, sort of like how I presented it today, that the more virus there is, the more the host has to throw immune activation at that problem to try to solve it. And I think that's the most simple explanation I have now, and until more data shows otherwise, I think that's a good one.
Yeah. Microphone number 2.
Rosalie Ponte from McGill University. I was wondering if in the HIV positive populations in the two groups, did you look at specific CD8 T cells, and would you expect to have the same results?
Yeah, there's a paper, I don't remember who the author was, but it was a blood paper, I want to say 2008, that did something similar. And that's one of the weaknesses in our study, is that we don't differentiate between the specific types, or the HIV-specific CD8 T cells. That's definitely on the wish list. And if their data, it falls in line with our cohort as well, I think we'd expect to see big differences. Perhaps that the HIV-specific T cells are the ones driving this effect.
Great. Well, thank you. We'll move on to the next speaker. Our next speaker is Wei Liu. Dr. Liu works as an associate professor in Peking Union Medical College Hospital, China. She is involved in infectious disease and studying immune pathogenesis and antiretroviral treatment in HIV. The topic of her discussion today is association between gut microbiome and CD4 recovery in HIV-infected patients.
Thank you, Alan. Good afternoon, everyone. It's my great honor to present our preliminary results on association between gut microbiota and CD4 recovery in HIV-infected patients in China. I don't know how to use it. Yeah. Since the first case was reported in China in 1985 at PUMCH, which I work for, the HIV/AIDS becomes one of the important public health problem in China. By the end of 2017, there were totally 758,000 living cases of HIV/AIDS in China, with more than 95% of sexual transmission. As we know, most patients who have successful viral load suppression and who can get very distinctively CD4 T cell reconstitution, but some individuals only have insufficient CD4 recovery, which are referred as immune non-responder, and many factors may contribute to immune non-responders, such as decreased thymus function, damaged bone marrow, some things. And when I did my review on those literature, and a question occurred to my idea: what's the relationship between microbial translocation and immune activation, and also with the CD4 recovery? So we performed a cross-sectional study on 4 groups during 2015 to 2016 in our hospital, and we recruited 26 HIV viremic untreated patients together with 30 healthy volunteers, and also we recruited immunologic ART responders, which are defined as those patients who were on ART more than 2 years and with CD4 T cells more than 350, and another group is called Immuno ART non-responders, which means they only got CD4 T cell less than 350 and on ART after 2 years. So this is the basic characteristics of this study population, and each subject has had a result of microbiota analyzed on metagenomic sequencing on the specimen of feces. Also, CD4 T cells subsets and immune activation subsets such as CD8, HLA-DR, and 38 positive markers were measured by flow cytometry. There were no significant difference in alpha diversity between 4 groups. Beta diversity was calculated by 2 measurements by principal coordinate analysis and nanometric multidimensional scaling, which showed a clear separation of health controllers from other groups at the genus level, which suggesting that those got this microbiota changes may only exist maybe due to HIV infection itself, other than antiretroviral therapy. also, we found distribution of those 4 groups in enterotypes, and untreated patients had the highest enterotype 2 microbiota, and those individuals on ART had a lower percentage of enterotype 2, which were predominant by Prevotella, still higher than healthy controls. This is the main part of the results. When comparing the gut microbiota change in immune non-responder and immune responder group, and we found those four species differ. Means those 4 species have relatively abundance in immune non-responder, and those 2, we can see this, and they were comparatively with CD4 T cells increase and decrease. And also these 2 species, they had a positive correlation with CD8 HLA-DR positive CD8 T cells, which is more interestingly that we also analyzed those untreated patients and comparing these 4 species in this population. Those patients who under treatment have have less than CD4 T cell, less than 300, also have the same abundance, relatively abundant species as in immune responder. So we get the conclusions. We confirm that gut microbiota abundance in enterotype 2 after HIV infection in Chinese population, and also the diversity and the composition of gut microbiota couldn't be recovered completely after ART. And also we found some gut microbiota was associated with poor CD4 T cell recovery and involved in immune activation, which might have effect on CD4 T cell since the beginning of HIV infection other than ART afterwards. So the intervention to reverse gut dysbiosis and inhibit immune activation might be new strategies for immune reconstitution in HIV. This was a cross-sectional study with limited number of patients. Now we are performing ongoing studies on a longitudinal study on naive patients before and after ART, and also we are going to do some animal models to confirm the results, and also gut biopsy are needed too. We are hoping to find some good probiotics for immune non-responder patients and to set up a clinical trial. And also we are performing a new clinical trial on Chinese herb and hope maybe they can have some impact of gut microbiota of HIV patients. Thanks for all my colleagues and who have contributed to this research. And you can see from the Chinese platform that through all Chinese government and the physicians, we were on the journey to ending AIDS in China. And thank you so much.
Thank you. This paper is now open for discussion. Microphone number 2.
Dr. Liu, thank you for a really great study. David Miller, United States. Is there any perceivable way to identify differentiations in presenting microbiota species that do recuperate in patients after antiviral therapy and before their infection?
I think it's a little hard to recruit those patients who just before infection, but after it's I think it's useful to make such trial on those patients who can after antiretroviral therapy. Yeah, but before infection, it's a little hard.
Microphone 3.
Thanks for presenting your data.
Thank you.
So it looked like on your table 1, where you're describing the baseline characteristics, your pre-ART patients, a substantial proportion of them had CD4 counts above 350. And you're defining CD4 response. I know it's difficult and many debate about how do you define CD4 responses, but if you restricted only to those individuals with a certain, CD4 less than 200 who responded to greater than 350, or looking at mean change in CD4 count. It seems like you're counting a third of your patients as responders even before you start ART.
Yeah, I think that's a good question, and I think because I have only 26
Yes.
patients pre-ART, and
But
we
maybe
are
looking at change in CD4
Yeah.
count or something
yeah,
like
that's
that.
why I'm doing a longitudinal studies now on 50 patients or 100 patients.
Microphone 2.
Hi, thank you. Adrian Eisler from Australia. I was just wondering, very, very interesting. I just wonder, on the subject of prebiotics and issues like, say, psyllium husk, just to give one example, do you feel there's any data yet to give any kind of evidence to substantiate that as perhaps one possible strategy for people like ourselves who might be able to sort of try and change the gut microbiome?
Yes, I think that's a good idea. And in fact, I think there might exist some prebiotics who can have impact on in CD4 recovery. But it's very hard to find some good prebiotic, which means accurate treatment for CD4 recovery. But I want to, we are just doing some animal models to confirm some bacteria from my study, and I think we can ask for some company to maybe to make some probiotics and to maybe to set up a clinical trial.
So you feel this may be an area that is worthy of further
I think
study?
So.
Okay, thank you.
Thank you.
Are there additional questions from the audience? Maybe I could ask one. One of the critical features of microbiome studies is clearly diet. And as you're thinking about in your country, in China versus other places in the world, and we try to compare data, what kind of information have you been collecting on diet, and how does that vary within the populations you're studying?
Yeah, that's a very good question because we have a different diet and also some different environment, some circumstances. But I think all my patients in my clinic, most of them live in Beijing city, and I think I only have Chinese population here, so I think that might be not a big confounder factor in my study.
But it'd be good to understand that as we're trying to compare around populations in the world, so we know what the baseline diets are in your populations as we look to other groups across the world.
So international cooperation are welcome, and just contact me with my email, just on the photo.
And go ahead, microphone number 2.
Just one more question, Dr. Liu. Has there been any contemporary consideration from investigators like yourself who are looking at HIV microbiome regarding fecal transplantation? Because that's what's typically been done with uninfected populations, and that's become to some degree standard of care now.
Yeah, that's my dream. That's my dream to do some FMT to treat HIV patients who, especially immune responder. But it's very difficult to pass the ethics committee, and also we need endoscopy, something like that. But anyway, I think that is the final solution to do something on this patient.
Yes, please use the microphone. Thank you.
Hi, I'm Howard Armstead, representing the Public Eye newspaper in Lesotho, and I'm 66 years old. I've been HIV positive for 34 years, and for 33 and a half, I participated in the MAX study at UCLA. I've been taking ARVs for 28 years, and I've been taking selenium for 18 years as an adjunct complementary therapy to ARVs. I took AZT for 8 years. I was probably on AZT longer than anybody, but I took it at a half dose. And if you go back and read all of the early literature on AZT, that was actually the best dose, 3 tablets a day. So I'm wondering, I've been to 27 international AIDS conferences, not all sponsored, some by the NIH, 2 mother-to-child transmission conferences, et cetera. And I'm wondering why there's never a session on selenium. Nobody can say selenium. Even when you raise a question about selenium, the people that answer cannot get the word selenium through their mouth, through their lips. So I'm wondering why the fact that selenium plays such an important role in not only hemorrhagic fever viruses like Ebola, et cetera, which I had the only effective treatment when they had Ebola. Why do we totally ignore selenium? And it's important to focus on the virology, but we totally ignore the immunology and how we can lift the immune system through complementary therapies. Well, I don't understand why nobody can get that.
I think that's a big question, and it's hard for me to
Yes.
to
That's
answer
a very big
it.
question.
But in my opinion, because I'm a physician and I don't care only about the virus and the CD4 T cells of my patients. I also care more about their life quality of long lifespans like this. So I hope we can do more things to help them.
Yeah, I just wanted to add, in 34 years of being HIV positive, I've never been sick, never had to go to the hospital, except when I was first infected about October 1983. And then I was sick as a dog, on a mattress, on the floor, sweating for a whole week. I thought I was going to die. And since then, I've never been sick ever, except maybe something like a fungal infection, jock itch or so. So I'm doing something very right as a long-term survivor, never being sick. And I think sometimes other people should take a look and examine somebody that's been very successful.
Yeah, I have been asked that question a lot, being a basic science researcher. And I think one of the answers to your question is where do we get funding to do some of this research? And I think if you believe strongly in that and we can advocate for funding for scientists, then we could perhaps do something like that.
The research has been done both as an early therapy without ARVs and as an adjunct therapy. Rwanda did great with, pre-ARV treatment, showed that 1 tablet of selenium could slow down progression of HIV becoming AIDS by 43.8%. And the Nigerian Institute for Medical Research, working with Harvard University, showed that 1 tablet of selenium, 200 microgram, added to heart therapy, more than double the increase in CD4 count and triple the increase in hemoglobin.
All right. Well, thank you again, and we'll move on. Thank you, Dr.
Thank you.
Our final presenter today, and then we'll move into an integrated discussion, is Clovis Palmer from Australia. He heads the HIV Immuno Metabolism Lab at the Burnet Institute in Melbourne, and lab focuses on how HIV regulates metabolic pathways and immune cells. Dr. Palmer.
Okay, so we know that naive or resting T cells, they generally are very metabolically less active than activated cells, and they preliminarily use glucose to generate energy via oxidative phosphorylation. What's interesting is that upon activation, whether or not it's due an infection or immune activation, there's a metabolic switch from oxidative phosphorylation towards glycolytic metabolism. And this is orchestrated by increased levels of glucose transporter 1 on immune cells, which are the main glucose transporters on immune cell. And so previous work from our lab shows that people living with HIV infection has higher level of glucose transporter 1 on their cell surface compared to those uninfected. And in fact, we saw that there was increased level of glucose uptake in CD4 from HIV positive individuals. Furthermore, we illustrated an increased level of lactate production in CD4 positive T cell, increased level of exokinase activity, PI3 kinase mTOR activity, and all of these illustrate the glycolytic switch. And interestingly, we also see that metabolically active cells expressing high levels of GLUT1 are highly infectious. And relevant to our observation, we saw that people living with HIV with low level of CD4 T cells have high level of glucotransporter 1. And as you can see from this graph here, there was an inverse relationship between CD4 T cell level and the percentage of cells expressing the glucotransporter 1, which is a marker of aerobic glycolysis. And clearly from this slide at the bottom right-hand side, you can see that HIV-negative individuals have relatively low levels of this glucotransporter 1 when compared to HIV-infected persons without therapy. But interestingly, when you look at immunological responders compared to non-immunological responders, you see an elevated level of glucotransporter 1 on these cells. So we wanted to gain a bit more insight into what's going on in these metabolically active CD4 positive T cells. So when we analyze the senescent cells looking at CD57 positive, CD28 negative cells, we clearly see an increased level of glucotransporter 1 on these senescent cells.
I'm going to ask you that question.
Yeah. And then looking further, we analyze exhausted cells measured by marker PD1 and expression. And clearly, as you can see again, there's increased level of glucotransporter 1 on these exhausted cells. So the take-home message here is that increased metabolic activation in these cells, increased level of glucose transporter 1 on CD4 cells is related to senescence and metabolic exhaustion. And so we developed this model, or proposed this model, whereby uninfected or resting CD4 T cells have low level of metabolic activity, that is low level of glucotransporter 1, glucose uptake, and low level of glycolysis. But during an infection like HIV, there is a metabolic reprogramming towards glycolytic metabolism. And what's interesting is that this metabolic reprogramming is less efficient than oxidative phosphorylation. And because CD4 T cells have low energy reserve, they are unable to withstand this metabolic or inflammatory insult, and therefore they are metabolically exhausted and they die off. So that's our working model. So how is glucose transporter 1 regulated? We know that glucose transporter 1 is sequestered in the cytoplasm of immune cells, and then when it becomes activated through the PI3 kinase mTOR pathway, there is a translocation from the cytoplasm to the cell surface membrane just to pull in more glucose into the cell to enhance aerobic glycolysis. And so the question we wanted to ask clearly was whether or not these genes or regulators that regulate glucotransporter 1 expression would be associated with CD4 T cell recovery, because here again we see that patients with low level of CD4 cells expressing glucotransporter 1, they are immunological responders. And so looking at this SNP here, which regulates glucotransporter 1 expression, we clearly see that the GG genotype, patient with GG genotype has high level of glucotransporter 1, and those with the GA and AA genotype has low level of glucose transporter 1 on their CD4 T cells. So if you look at this table, you clearly see that the GA and the AA genotype were associated with immunological responder. So if if anybody living with HIV, they would want to be the GA and the AA genotype. And so our concluding points and significance is that metabolic reprogramming of CD4 T cells from oxidative phosphorylation towards glycolytic metabolism, which is a less energy efficient process, is associated with low CD4 T cell count. And the question emerges now whether or not immunometabolic targeting of these cells by targeting the PI3 kinase, the mTOR pathway, could be a new therapeutic strategy to improve immunity and recovery in HIV positive individuals. Data that I haven't presented here show that this metabolic shift from oxidative phosphorylation towards glycolysis causes this inflammatory phenotype. So these CD4 cells produce more TNF, interferon gamma, etc. And this was similar to monocytes, which I won't go into now. Thank you for your attention.
This paper is now open for discussion. Are there questions? Yes. Microphone 2.
Hey, Clovis. Thank you so much, because this is very insightful regarding immunometabolics. Quick question in regards to this. Is there any way to identify whether the characteristics you presented here may also be reflective of what's going on in Th17s in the gall tissue?
Yeah, we didn't specifically look at Th17.1, but as I alluded earlier on, is that we saw this metabolic increase in Th17 cells in general and in inflammatory subpopulation. So when you look at Th1 cells, for example, you see this metabolic shift towards aerobic glycolysis. Again, this was similar to monocytes inflammatory subset. The intermediate subset were highly glycolytic, and they were more pro-inflammatory.
Microphone 2.
Really nice, Clovis. You have a unique opportunity, it looks like, if you have a genetic predictor of a biomarker, then to take it the next step further and look at events and Mendelian randomization type studies where you can get a little bit more causal inference than is typical in observational studies.
Yeah, we were just having a chat with Rena, one of my collaborators, sitting right around there, and she has a very nice cohort in Malaysia, and it's something that we definitely going to be looking at.
Great.
Yeah.
Would you care to comment? You mentioned therapeutic targeting. Have you had an opportunity, as you said, the mTOR pathway is one that we target with rapamycin and other drugs. Have you had a chance, and there are studies in HIV where we are using the drug and have to treat patients in some of the cure agenda studies. So have you had a chance to look? And that may also be critical as we're trying to look at these new drugs, given the genetic polymorphisms of being able to have those genotypes done on those patients ahead of time in terms of the responsiveness.
Yes, so I know that SOX and Stephen Dix in San Francisco, they're doing some work looking at sirolimus. I think everolimus as well, which are mTOR inhibitors. So we are actually going to piggyback on their studies to see whether or not we can identify if these drugs was able to suppress inflammation and improve CD4 T cell count.
Yeah,
but we have also looked in vitro with other more targeted approach. So we look at PI3 kinase gamma inhibitors, which are more specific towards T cells, and we found an incredible observation where these cells suppress immune activation in culture, and they prevented these cells from dying off in culture as well.
And just one other bit of information that may be helpful. Mark and Cheryl Cameron are at a recent AIDS clinical trial group meeting presented data on RNA profiling showing mTOR as a marker in the CLIF study for immune non-responders. So I think that may be another relevant cohort to speak about, because again, if that may be then confirming some of the pathways that you're looking at. Microphone number 2.
Actually, my comment is related to those trials. I was wondering if you are aware of the trial on metformin, and if you could comment on that. To my knowledge, it's like the first trial showing a destabilization of the reservoir by the group of Dr. Ndlovu in Hawaii.
Yeah. So I know that metformin is a kinase activator, which indirectly suppress mTOR. So I definitely think that metformin could have potential in reducing inflammation and potentially improve immunity. In fact, there are several studies showing in aging individuals given metformin, they increase the efficacy of flu vaccine. It's clearly illustrating that metformin can improve immunity. Combination of metformin and mTOR inhibitors like sirolimus, everolimus, I'm not sure. That's definitely some studies that needs to be done.
Microphone number 2.
Clovis, there have been 2 studies presented here on the use of naltrexone and low-dose naltrexone, which shows there's some pretty serious consideration for its anti-inflammatory properties. Do you think it would have any potential utility for addressing a study for immune metabolism?
I'm not sure. I'm not with regarding to the specific pathways that that drug target, and I think that's one of the important things we have to consider when we are doing these clinical trials. Is look at a more focused approach. Make sure that we know exactly what these drugs are targeting. That we just don't include patients in these clinical trials just for the sake of it, but we actually know what we are getting into. And so I think that that's important, not to just fish around, but actually ask specific question using specific pathways.
Clovis, a lot of the interest in HIV is at the tissue level. Have you had an opportunity to look at the glucose metabolic profiles of cells either from gut tissue or lymph nodes?
No. Well, we actually started that in your lab, but as you know, it's very difficult to get these tissue samples, and some of the processes involved in actually extracting the cells from these tissue could affect the metabolic activity. So whilst I'm aware and I'm sure that there's something happening, I think we still need to perfect the methods for extracting these cells.
Right. Well, I'd like to thank Clovis, and I'm going to then open this up. We have a few minutes left for general discussion. Are there some other general comments or thoughts? Maybe I can start and ask about whether or not Anthony might like to comment on the role of telomeres in immunometabolism. And sort of, I know people may not have specific data, but at least maybe some speculation on where we might be able to integrate some of the topics here today, and also on some of the glycan work, because I think from each of the presenters today there are some all four of us, you could do some integrated studies together to understand aging and immunity.
So, okay, I'm going to take a more broad approach first. In population studies, when we look at telomere length in whole blood, we'll see invariably a decline with age if the range of age is large enough. So there's something within the cells of the blood that's driving that, but we don't know exactly what that is. And there are a lot of, well, not a lot, but when doing seroconversion studies as well as ART initiation studies, sometimes people see an immediate increase or an immediate decrease of LTL, so leukocyte telomere length, happening at such a rate that makes me think it's not the same. It's not the same effect as the linear decline with age. So what we probably want is a composite between the telomere length of some immune subset that reflects aging the most and the quantity of immune subsets, of certain immune subsets like the CD28 positive or negative. And that second part, I think, is where the metabolic data would come in. Once we find out which subset proportions are the most representative of that long-term decline with age, then we can look within those populations and do the glycan work and do the telomere work there.
It's fascinating. In fact, our lab is quite interested in aging, and Susan Crow has done a lot of work looking at monocyte aging in HIV infection, and she with Anna Herbst actually did some work with telomeres as well. And so one of the questions we wanted to ask is the relationship between metabolic dysregulation and aging, and I think there is growing consensus in the literature illustrating that there is this metabolic dysfunction and metabolic shift towards aerobic glycolysis in aging cells. And to what extent that happens in the context of HIV is still yet to be defined. And so that's one of the area of work that we are really interested in. We haven't really delved into the T cell as yet, so I'd definitely be interested to chat with you to see what we could do together.
Yeah. The key, to reiterate, is to find which cell subset to look at. I think if we look at all lymphocytes or leukocytes, the data will be very noisy. Yeah.
Well, isn't there a flow cytometric method available so you could identify cells by that?
So, Chiara, would you care to comment on the role of glycans in aging and maybe how it might relate to the microbiome? Because I think there's also an important role for, you talk about the host, but also we have bacteria So maybe we could think about what would you think about the microbiome and glycan biology?
Well, it's a hard connection because to date, as far as I know, there are no studies that relate these topics. But you know, the glycans derives from circulating immunoglobulin. So some relationship may be find if we look at also at the immune system in the gut. So.
Why would you?
Yes, for the first thing I want to say that I think in China now we have the big problem of late presenters and together with aging new reported case. So I think we need more analyze and research on those aging things, especially with some metabolic syndrome, many comorbidity disease. The second thing is about microbiome research, and as you know, it costs a lot and it costs time-consuming research. So we need more incorporation with all of you, and we need help. And we want to send our new era students to your country, to your esteemed laboratory to learn more. Thank you.
Any final comments, Clovis, at your end?
I guess one of the things that I'm a little bit a bit concerned and with our field is that I think we're not moving fast enough in this area, and I think we need to adopt and develop new technologies and approaches. And I know that, for example, in the field of immunometabolism, it has really changed how the immunology field in general, and many areas, example cancer and diabetes, for example, has really taken on the immunometabolism, metabolomics field. And this is really lacking in HIV. I mean, it's like we are recycling the last 10 years of basic data in our area. And so therefore, I think to solve some of these problems and to answer critical questions to help patients, we need to come out of our box and take in more information.
Yeah, I think we heard that from this morning's plenary speakers, that we really need to cross the borders, as we've had here today from all the different groups in Canada, Italy, China, and Australia. So I think this is really a very good example of how we can get cross-country and get investigators to really work and use technologies from other fields. So I'd like to thank all the speakers and thank all the audience for their attention. Thank you.